organization, the levels of organization, and I haven't finished this. Let me go through the size of the specimen. Now here in the test, they always get you a picture, for example, like this, like a mitochondrion or whatever, and they ask you to get its actual size, for example, or they ask you to get the magnification. So we always use the rule magnification equals the image size divided by the actual size. Suppose they gave you the magnification, for example, times 400. So this is easy to get the actual size because you have already the magnification and you have the image size. Now, how can you get the image size, although they didn't write it in the question? You just get your ruler and measure from the widest points. Never measure like this, for example. This is not the widest. The widest part of the picture, you are going to put your ruler across it and you'll measure, of course, in millimeters. Now, in OL, we always use the millimeter and the micrometer. We never use a centimeter or the nanometer. So let me go through these measurements. We have 1 millimeter equal 1000 micrometer. So when you measure with your ruler, you put your 0 point here and you get the measurement in millimeters. Suppose you have, for example, 3 centimeters and 0.6. So this is, of course, going to be 36 millimeters, and you just change it to micrometers. Suppose they gave you a measurement by a micrometer. So 1 micrometer equal 1/1000 of a millimeter, or we can say 10 to the -3 of a millimeter. Type. This means that you have already the image size. You are going to measure it. They gave you the magnification. Then it's easy to get the actual size. But how can I get the actual size from something like that? You're just going to put the triangle, because some of the students, they get mixed up with this, and you are going to say I am in reverse. I am. So here is the image size, the magnification, and the actual size. If you want the magnification, you just divide the rest, 1 over the other, like this. So here I have I over A. And this is the original rule. Suppose we want the image. So you just multiply what's found here. So the image size is magnification times the actual size. But suppose you want the actual size, just divide the others. So here I will put the actual size equal image divided by magnification. So if they want the actual size, you just divide the image by the magnification to give you the actual size of the image, of this image. Sometimes they need the magnification. So of course they are going to give you the actual size. Sometimes they do not give the image size because you can measure it. And whenever you write the magnification, please write the times at the beginning. We say 400 times, but we write the times at the beginning. So that's all about the size of the specimen. And let me go through the cell bank. So what is the cell? We say that the cell is the unit of function and structure of the living organism. So what does this mean? It means that I said that it is the unit. It means it is the one part. If I added several cells together, they are going to give me the whole organism. So what about structure? The unit of structure. It means that if I gather, of course, thousands or hundreds or even trillions of cells together, they are going to give me a structure of the living organism. So what's the function? Every group of cells, they have a certain structure by which they can perform a function. يعني maybe we dealt with this last time in لما بيبقى في undifferentiated cells or unspecialized cells. So they take a certain structure, means that they differentiate in order to be specialized to do a certain function. But whenever we have a group of cells, similar cells, they are differentiated the same way and they are gathered together, this will give me a tissue, and the tissue, different tissues will give me an organ. Now, how do these different tissues work together within an organ? We say that these different tissues, they work in harmony together in order to allow this organ to perform its function. But what's the unit of the organ or the whole organism? It is the cell. Because the cell differentiated, so it allowed the whole organ to perform a certain function. So we say that all the cells, they are capable of doing all the vital activities of the living organism. So what's the meaning of the vital activities? It means everything, all the processes that are going to be done in order to keep this organism alive, because the word vital is coming from word vie, in French means life. So the activities or these processes that are done by each group of cells, if we collected all the activities together, so this person will remain alive. Now, we have two types of cells. They classified them again later on into other classifications. Later on in AS, you are going to deal with this. We have two types of cells. We have the prokaryotic cells and eukaryotic cells. And as I mentioned last time, that the difference between both is that the prokaryotic cells, they do not have a true nucleus, and they do not have most of the organelles, or nearly no organelles. Nearly. Yani the only organelle that is found in the prokaryotic cell is the ribosome, which is responsible for protein synthesis. But, and of course, the ribosomes, they differ a bit from the ribosomes found in the eukaryotic cells. While here they have a true nucleus together with all the organelles. So that's the difference between the eukaryotic cells and the prokaryotic cells. Of course, we can never see these cells by our naked eye, but we have to use a microscope. So we have two types of microscopes. We have the light microscope, and we have microscope, and we have the electron microscope. So what's the difference between both according to what we see? In the light microscope, we see that it allows us to see the gross structure of the cell. But here in the electron microscope, it allows us to see the ultra structure of the cell. So what's the meaning of the ultra? Means the detailed structure. So for example, if I looked at a plant, at an animal cell by using the light microscope, I'm not going to see except the cell membrane from outside. I'm going to see a dark shade, which is the nucleus. And if the microscope is very good, I can see other shades which represent the mitochondria. Of course, the mitochondria, they are the organelles that are responsible for aerobic respiration. That's all. And we are going to see the cytoplasm, like, for example, as if there is something cut into pieces like this, or we say that it looks like ground glass or smashed glass. But in the electron microscope, you can see every detail inside the cell, and even inside the organelles, you can visualize these. Like, is this the only difference between the light and the electron microscope? Of course not. In the light microscope, we use light as a source of energy, but in the electron microscope, we use electrons or electron beams. We use here for the light either the sunlight, or we use artificial light, or like an electric light, inbuilt light in the microscope. Here in the electron microscope, we use something called the electron gun that's going to produce electrons, and we use these electrons to visualize a detailed structure of the cell. In the light microscope, we use lenses, and these lenses, they are made of glass. But in the electron microscope, we use electromagnetic lenses. So what's the meaning of electromagnetic lenses? It means these are lenses that are going to attract, like a magnet, they are going to attract the electrons to allow them to fall on my sample in order to be able to see them. A light microscope, a best light microscope, will maximum magnify the image 1500 times, but here it can magnify from 100,000 to 500,000 times. And there are some electron microscopes that can magnify to 1 million times even. That's all between the difference between the light and the electron microscope. And let's go now through the cell structure that is seen either by the light microscope or the electron microscope. For both animal and plant cells, they have a cell membrane. يبقى common structures between animal and plant cells. We have a cell membrane, and by the way, it has two other names. So if you saw both names, other names in the test, don't be confused. It's called the cell surface membrane or plasma membrane. All of them give me the same meaning. A cell membrane or the cell surface membrane, of course, it surrounds both animal and plant cells. In order to, suppose this is an animal cell, this is the cell membrane that encloses all the structures or the components of the cell. Enclosing all the components, and also it's going to prevent leakage of the cytoplasm or any other material that is found inside, or any other organelle, because if the cell membrane is damaged, so the cytoplasm will leak out, taking with it some of the organelles. There is a very important function for the cell membrane, which is selective permeability. And sometimes we call it partially permeable. So what does this mean? Partially permeable, it means that it will pass some materials to the inside of the cell and allow some materials to get out of the cell. Of course, the materials that will pass inside to the inside of the cell are the materials that the cell needs, and the materials that will pass out of the cell, they are either wastes or materials in excess. Sometimes the materials inside the cell, they are not waste materials, but the cell does not need them. That's why they have to get out. The cell membrane selects which materials to enter and which materials to exit. That's why we say that it has selective permeability. But what is the cell membrane made of? The cell membrane is made of two components. The main component is a kind of fat called, of course, the phospholipid. This is a kind of lipid or a kind of fat, and it's made of proteins. Of course, there are other components for the cell membrane, but you don't have to know them in OL. But this is about the cell membrane, whether in animal or plant cells. This is a common structure between both. The next common structure is the nucleus. Here, the nucleus, when I was telling you about the light microscope, I told you that we can see the nucleus by the light microscope. Now, why can we see it? Because it is the largest organelle. And the word organelle, of course, means a minute organ. So this is the largest organelle that is found inside the cell. The nucleus is very, very important, and it controls everything dealing with the cell because it carries inside it the DNA molecules. And on these DNA molecules, there are genes. And these genes, they control everything in the cell or even in the whole organism. But the genes, they control all the activities of the cell, whether cell division, chemical activities, whatever. And also these genes, they carry our genetic information. That's why the nucleus is very, very important. It is the manager of all the cell because of the presence of these genes of the DNA, and these genes, they are responsible for controlling all the processes that the cell is going to do, or and because, of course, it carries the genetic information. Here, these DNA molecules, in a certain stage of the life of the cell, they form the chromosomes. So what are these chromosomes? Chromosomes, they are, if I have here the nuclear envelope, a true nucleus for eukaryotic cells, or even if a prokaryotic cell, I'm going to explain this now, we have the chromosomes inside looking like thread-like structures inside the nucleus. Now, these chromosomes, they can never be seen by the light microscope and even by the electron microscope, except when the cell starts to divide. Yani, if the cell is not dividing, we can never see the chromosomes. When the cell starts to divide, in order to give me 2 cells or 4 cells or whatever, so we can see the chromosomes. Here, these chromosomes, they are inherited from our parents. Yani, suppose, for example, somebody, this is the father that produces sperms, and this sperm, it carries chromosomes. And of course, if this is a mother and her egg cells, one egg cell even, will carry her chromosomes, the chromosomes of the mother. So when the sperm fertilizes the egg cell, both chromosomes, they go to the zygote. It means that this zygote, which is going to be a living organism later on, it carries chromosomes from both the father and the mother. That's why we say that the chromosomes are inherited from our parents. If the cell is not dividing, we can never see the chromosomes because they appear as how they are in the form of long, very, very thin threads, can never be seen even with electron microscope. If the cell will begin to divide, so these chromosomes, they are going to be thicker and shorter. So because they became thicker, if we stained this cell, we can see the chromosomes even by the light microscope if it is good, of a good quality. But when the cell starts to divide, the chromosomes become thicker and shorter. And we say that the chromosomes, they become condensed. But if we stained or gave a color to this, for example, a sample or a slide or whatever, we can see the chromosomes if they are condensed or if the cell is beginning to divide. But what about the other main component, or a common component? Cytoplasm. Cytoplasm is a jelly-like fluid that is found inside the cell, and it looks like the egg white before cooking. Most of it is made of water. But a cytoplasm is a form of solution. Most of it is formed of water. Nearly 72% of the cytoplasm is formed in water, in which there are solutes dissolved in this water. But these solutes can be one of two kinds. Of course, chemical materials, either inorganic solutes, like, for example, sodium, potassium, chloride, whatever, nitrates, and so on. All these are inorganic chemicals, or organic chemicals, like, for example, glucose, like amino acids, and so on. But because the cytoplasm is formed of water, in which there are solutes dissolved in this water, so the cytoplasm is a kind of solution, but because of the density of the materials that form this solution, so that's why the cytoplasm appears as a viscous fluid. We call it a viscous jelly-like fluid. Of course, viscous, as if it is sticky, because of the presence of these solutes that give a certain density to the solution of the forming the cytoplasm. The main substance, or the most important substance that is found dissolved in the cytoplasm are the amino acids, because as I told you last time, in the amino acids, they will form proteins, and these proteins are responsible for every possible process that happens in the body. For example, because the proteins, they form enzymes. The enzymes are responsible for all the chemical reactions, speeding up to the chemical reactions inside the cell. That's why these are very, very important organic molecules that are found dissolved in the cytoplasm. And because the cytoplasm contains these chemicals in the presence of water, we say that the cytoplasm is the site of metabolic reactions. So what is metabolism? Metabolism, this is a group of chemical reactions that are either building up or breaking down. Building up means anabolic reactions. we get small molecules like amino acid molecules, and we build large molecule, which is protein. If we want to break down this protein into amino acids, so it's a breaking down metabolic reaction, or it's called a catabolic reaction. So metabolic means, anabolic means building up. It means that we have small molecules like amino acids, for example, they are going to build up large molecule like protein. Catabolic is the opposite. It means breaking down. Means that if I have a large molecule like protein and I want to break it down into its components, so this is a catabolic reaction that changes protein into amino acid. Both reactions, they need, of course, enzymes. And these enzymes are found inside the cell. These are common structures between animal and plant cells, and there are other structures as well, which are the organelles, but I'm just giving you the main structures. Let me give you another main structure, but it's only found in the plant cell, which is the cell wall, of course. In plant cells, the cell wall is formed of cellulose. Of course, plant cell, the cell walls in bacteria differ from those of the plants, or differ from those of the fungi, and so on. As I told you last time, that the kind of cell wall differs. In bacteria, it's made of a material made of something called the peptidoglycan. It's something like a polysaccharide and a protein, where for fungi, it's formed of chitin, which is a polysaccharide together with the lipid or fat. Here, the cell wall of the plants is just formed of cellulose, and this cellulose is made of cellulose fibers. So these fibers, when they are arranged together like this in different directions, so they give me the material of the cell wall, as I'm going to show you here. Where is it? Just a second to get the folder. Here it is. Give it a few seconds to upload. Yes, here it is. So if you can see here, don't look at this part. Here are the cellulose fibers. They are arranged in opposite directions like this, and this is an actual cell wall. Look how it looks.
Yes, they're crisscross over each other.
Yes, perpendicular like this. And when you go, inshallah, to AS, you're going to learn this in details, but not in OL. This is the material of the cellulose or the cell wall. It looks like this, as if it looks like a piece of cloth. What's the function of this cell wall? This cell wall, it has three functions. It gives the cell, the plant cell, of course, its structure or its shape. Because when you look at an animal cell, it doesn't have a definite shape. But the plant cell has a definite shape because of the presence of this. Sometimes they look like this, to be hexagonal, for example, because of this cell wall. It gives protection. Of course, it protects the materials inside the cell. And there is a very, very important function which you always forget: turgidity. So what's the meaning of turgidity? Turgidity means that if the plant cell absorbs too much water, so the plant cell will not burst. Of course, to a certain extent, because this cell wall is giving it, is preventing bursting of the cell. if an animal cell took in or absorbed too much water, there is no cell wall to protect it, and cell membrane is very weak and delicate, so the animal cell will burst. But the plant cell, no. Instead of looking like this, for example, a plant cell, so if it took in too much water, it's going to look as if swollen like this. This is called turgidity. And by the way, there is another factor or organelle that helps turgidity, which is the vacuole that I'm going to explain now. The material of cellulose is somewhat rigid. Yes, it is not like wood, but it's somewhat rigid. That's why it gives, or it prevents the bursting of the plant cell when it absorbs too much water. And by the way, this cell wall is permeable to anything. We say that it is fully permeable. It means that it can pass everything, but immediately just next to the cell wall, sticking with it like this, is the cell membrane. But I have just said that the cell membrane is partially permeable. It means that the cell wall is going to pass everything, but the cell membrane will say stop. Only the materials that are needed are allowed to pass. Cell membrane, by the way, the plant cell, if you looked under the microscope, you can never differentiate the cell membrane from the cell wall because they are sticking together. They are very, very near to each other. We can never distinguish between the cell membrane and the cell wall in a plant cell while using the light microscope. But of course, we can distinguish between them when we look through the electron microscope. طبعا this cell wall, in industries, they get use of this cell wall because paper, for example, is made of cellulose. So they use the cell wall. But what about us? Yani, if we humans, if we eat plants, for example, fruits, vegetables, whatever, this material or the material of the plant cells can be digested, except cellulose. Cellulose cannot be digested inside our intestine, and they change into fiber. I'm going to re-explain this when we revise digestive system. Change into fiber inside our large intestine, and this is very healthy, by the way, to the large intestine. I'm going to explain this in details after when I explain human digestive system. Now, these were the common main structures between the animal and plant cell, and the cell wall is only for the plant cell and not in the animal cell. But what about other structures that are common and what are found only in plant cells and not in animal cells? Let's go to the common organelles between animal and plant cells. There is a very important organelle, طبعا, the mitochondria. Mitochondria, these are very small organelles, but they, if I compared the size of the mitochondria with other organelles, this is very large. This is the second largest organelle after the nucleus. And it is found in eukaryotic cells, never in prokaryotic cells, and it's seen by the electron microscope. Just a second to get this folder. It can be seen in details by using the electron microscope. I'm going to show you a hand-drawn picture for the mitochondria, just to show you the gross structure of this mitochondria, of the detailed structure, I'm sorry. And it nearly looks like this is a hand-drawn picture, and it is only seen these details by the electron microscope. If you have a very good light microscope, you can just see a shade, as I told you before, for the mitochondria. Now here, this structure is responsible for aerobic respiration. Means respiration in the presence of oxygen. Of course, when the mitochondria utilizes glucose together with the oxygen, so it's going to give me at the end energy, because from our previous studies we know that respiration is glucose. When it is broken down or oxidized in the presence of oxygen, this will give us energy by which we do all our activities, plus carbon dioxide, of course, plus water. I shouldn't write symbols with words, but just to to save the time. Because this mitochondria, we call it the powerhouse or the organelle responsible for producing energy, so its number is too much in the active cells. Do we have inactive cells? No, all the cells are active, but we have active cells and we have less active cells. The active cells that are found in our body are the cells of the liver, cells of the muscles, the brain cells, the sperm cells. These are active cells. But what about the plant? Does it have active cells as well? Yes, of course. The cells that produce photosynthesis, which are called the Palisade cells. These are active cells as well in the plant body. Now, what about the less active cells? Any type of cell, blood cells, for example, white blood cells, whatever, the bone cells, the cartilage, all these are less active cells. They contain mitochondria, but the active cells, they produce more, or they contain more mitochondria, because the active cells, they need more and more energy. That's why they have to include more and more mitochondria in order, of course, to perform their function. Now, I'd like to mention something about the animal cells. They have certain granules that are called the glycogen granules, and they appear like black spots in the electron micrograph or the electron microscope image, and these are reserve fuel. If, for example, I get the board, I just said that glucose is needed to produce energy. Suppose this glucose, there is a shortage of glucose in the body cells, so there are reserves called glycogen grains that are found inside our cells, and this glycogen can change into glucose in order, if we needed extra glucose in our cells. Of course, I'm going to explain this glycogen when I explain molecules of life. Here, the first common organelle is the mitochondria. The second common organelle between the plant and animal cell, and even it is found in the prokaryotes like bacteria, the ribosomes. Ribosomes are the smallest organelles. You can never see them by the light microscope. and even by the electron microscope, you must need a very good electron microscope that has a very high resolution power in order to show you the ribosomes, because they are very, very small. These ribosomes are responsible for production of protein. That's why I told you they are very important. That's why. It's not the ribosomes that are important. It is the material that they produce, how they synthesize. The proteins are the most important molecules of life. As I always say, they are responsible for our life. That's why we call them molecules of life, at least because they form enzymes. And without enzymes, we cannot keep on living. Enzymes, they are proteins. That's why we say that proteins are the molecules of life. For ribosomes, we always say they are, although they are very, very small, but they are of the most important organelles in the body, cells, because they produce proteins. But let me go now to organelles that are found only in plant cells, not in animal cells. Of course, I mentioned before the cell wall. This is called a structural a component of the cell. But if I am explaining something about the organelles, because the organelles, they do a certain function, so I say that they are the functional components. The structural components means the components that give structure, like the cell wall, the cell membrane, the cytoplasm. Yes, they have different functions, but they give structure to the cell. Functional organelles are the organelles that perform a certain function, like ribosomes, like mitochondria, and they are found in common with animal and plant cells. But here in plant cells, we find, in addition, vacuoles and, of course, chloroplasts. So as for the vacuoles, if I have a plant cell like this, then the vacuole is occupying most of the cell. And it is found very, very large, in a very large space inside the cell, and it is pushing all the other components to one side. Now, what is this vacuole? A vacuole, we say that it is a membranous sac. Membranous sac because it is covered by a membrane, and this membrane is called the tonoplast. Inside the vacuole, there is the cell sap. And I remember I explained this last time when I was explaining the plants. A cell sap, it is just a solution that is
containing
found.
water and nutrients,
Yes,
sugars, salts, and pigments.
very good, together with waste and so on. It is only found in the plant cell. And I mentioned last time that the animal cells, they have vacuoles, but they are called vesicles. And I told you before that the vacuoles, they are permanent, but the vesicles, they are temporary. What about the chloroplasts? The chloroplasts, of course, they contain chlorophyll. And their size is just greater than the mitochondria. There is a very, very little difference between the chloroplasts and the mitochondria, and the chloroplasts are bigger. Because they contain chlorophyll, so they are responsible, of course, for photosynthesis. Inside the chloroplasts, just a second, I'm going to search here for a chloroplast. Where did all these organelles go? Because I updated the pictures, I removed some of them, but they are found على PowerPoint. Here, just let me return back to the chloroplast. Chloroplast, it contains chlorophyll that's responsible for photosynthesis. Now, if I have a chloroplast like this, together with the materials or the components that contain chlorophyll, these look like disc-like structures like this, and they contain chlorophyll. We find here certain grains, and these grains, they become full of starch, because after photosynthesis forms glucose, this glucose is going to change into starch in order to be stored in the chloroplast. Starch grains, they are found in the chloroplasts as storing grains for starch. If the plant needs extra glucose, so this starch is going to be broken down into glucose once more in order to be used by the plant. These are the structures that are found in the plant cell only and not in the animal cells. I would like now to give you some questions about chapter 1 and 2. And let me open this. I prepared some questions. Past papers, of course. Let me search for any question dealing with chapter 1 and 2. This is a food chain. No. Because oh yes, here it is. I always look for very old questions because they repeat the questions from very old exams. These are cunning. I found that they get questions from 2005, 2007, and so on. Let us read this question, number 6, and answer it, please.
The answer is D, reptiles.
Yes, very good. Now let's go to the next one. This is dichotomous key, of course. I know the snail is not in the new syllabus, but there is a picture that you can rely on.
Is it animal C?
Yes, very good. Now let's go to, now these are enzymes. They are not putting, in the old exams, they do not put them in order. In new exams, they put them in order. Water pass, no. Quarter, no. No. I think that's all with this. It's chapter 1 and 2. Yes, here is another dichotomous key. But let me zoom this out a bit. Here.
B, 2?
2, of course. Of course, you're relying on counting the legs.
Number of legs, yes.
Yes, yes. Although this lice, or pediculus, the louse, its scientific name is pediculus, they always say, most of the students, they say that this is an arachnid. No, it has 3 pairs of legs, not 4 like this. This is a crustacea, of course. Now let's go to search for another one. For new exams, it's good. يلا number 22 because they put them in order. Number 22.
D, sensitivity.
Yes, of course. Yellow number 27.
Q and R.
Very good. Type number 28. Palisade cell, of course, it's a plant cell, as they just said. 28.
D, many chloroplasts.
Very good. No. That contain breakdown products, bacterial cells. No. I think that's it. What is this? Never mind about this. This is chapter 13. Number 38. Of course, this is something from yani, coming chapter. Yes, of course, you know it.
Cytoplasm?
It's not common in all of these cells. And you have taken this before. What's not common?
Right. What's not common? A diploid number of chromosomes.
Yes, of course, of course.
I thought it was common. I read common.
No, not common. خلاص, that's it. Let's go to another one. This is 2011. Yes, number 1.
Sensitivity.
Yes. What about number 2?
Myriapods, crustaceans, arachnids, insects. D.
Very good. Number 3. Annelid is a kind of worms. You haven't taken this. لا خلاص, it was cancelled. Number 4.
Cell wall?
Yes, of course. Number 5.
Central vacuum.
Central vacuole, yes. Position of the nucleus is correctly. طيب, okay, number 6. Of course, this is a plant cell.
D.
Number.
Actually, or B?
Which cell shows the position correctly?
B.
B, yes. 7.
C.
C, excellent. Let's go to another one. No. I think that's it. No, so let's go to another exam. That's the one. Yes, number one. Let me read it in the beginning.
It can already make, okay, it's C.
C, very good. Number two.
C.
C, very good. Number three.
B.
B, excellent. Yes. Number four.
D.
Okay, you have here the dorsal fin.
Yeah, is it this black spots one?
It
Or,
is. These are black
okay,
spots, two
yeah.
of them. يبقى. And this is another one.
I based it off what is under, like exactly where they arrowed the dorsal fin, that's why I said.
No, the dorsal fin takes from the beginning till the end like this. So this
So
is
it's
number
C.
C, of course. No. Yes, number six.
A chloroplast.
Of course. Number seven.
B.
B, of course. Number eight.
B. A.
Here is correct.
What? Is correct.
Is correct.
Um,
Take care because these are very much confusing. I know.
C.
C, of course. Thank you. Yes, number nine, temperature increase. No, we haven't taken this yet. Which direction? Actually, no. No, that's all. Let's go back to theory questions. Yes, read this question and start.
I'm gonna say they have an abdomen.
An abdomen.
Do I say many pairs of jointed legs?
No, you can never say many
Because
pairs.
there's
This is a scorpion, so you have
yeah
to know how
four
many.
yeah
Four
four
pairs.
yeah
Yes,
they have four pairs.
yes.
Crustaceans also have more than four pairs.
Mm-hmm.
Myriapods more than four pairs also. We cannot count. And our insects I have only three, so I cannot say that, right?
They must be visible. Okay. You can tell me what are the visible three features that you can see in front of you that arachnids show with other
Yeah,
arthropods.
yeah.
You said that they have four pairs of jointed legs, while others have طبعا different numbers.
Yes.
What about the rest of the features? I want to.
Uh, Here, arachnids, which is the scorpion, it has simple eyes, which is not the same as the others.
But the myriapods, they have simple eyes.
Yeah, but crustaceans have compound eyes and insects have compound eyes.
Okay, but I want the same things that they share. Yani they share jointed
Oh,
legs, yes, because the jointed, the word joint.
okay.
What else? You mentioned something about the abdomen.
Yeah, I said they also
Describe
have
it.
an abdomen.
Okay, describe the abdomen for the three or for the other three as
Okay.
well.
Well, the arachnids are divided into two regions: the cephalothorax and abdomen,
Mhm,
and myriapods are also have an abdomen also,
mhm.
and crustaceans they don't have any.
So why crustacea they have? They have cephalothorax and abdomen. So
Okay, so then they
why don't
share.
you say segmented body?
Oh yeah,
That's it, segmented body with jointed legs and what else? Can you see in
jointed
front of
legs, segmented. I cannot say antenna because arachnids don't have
you,
antenna.
of course?
I don't know what else could it be.
What about the covering that you can see in front of you?
Oh, exoskeleton.
Exoskeleton, yes, they share this together.
Exoskeleton.
Let's go to next question. Let me sort this out a bit. What is this? Let me go to the question and then I return it back. Oh, this is the dichotomous key. Then I'm going to raise the picture every now and then. I will scroll up and down. Let us see the first statement. Abdomen without a tail. Then
Yes.
this will be,
Do I just choose any of them?
excuse me,
Do I just choose any of them?
you have here,
It says
it's an
abdomen
abdomen
without a
without a
tail.
tail, so go to 2.
Yeah.
In 2, legs much longer, then go to 3. And you are going to begin
Yeah, yeah,
from number 3, hairs on the legs. But you have to
yeah.
choose the ones that have abdomen without a tail, legs with much longer than the abdomen, and
Mm-hmm.
cephalothorax, legs are not much longer than the abdomen and cephalothorax. This is very absurd that they put every part in a page. Uh, we can never memorize what's written. Hairs on the legs. So let us see, where are the hairs on the legs? Which one?
A has
A,
hairs
of
on the
course.
legs,
Yes, of course. So
and the legs are longer than the cephalothorax, so that confirms.
yes. Now number B, no hairs on the legs, but they must have the three descriptions or
Yes,
the two
yes,
descriptions above.
yes.
No hair on the legs.
So it's gonna be E.
E. But look at here. They said that the legs are much longer
Oh,
than the
much longer.
abdomen.
I thought just longer, longer. Okay. It's gonna be this one. G.
G, yes. Mm,
Wait, so it's that small part is just a cephalothorax?
the small parts, yes.
See, I can barely see its body for
Oh,
G.
for G. G, they said that there is no hair on the legs,
Yeah.
and the legs are much longer in the than the abdomen. Legs are much longer than the abdomen and cephalothorax. Not much longer. Okay, now number 4A. Cephalothorax or abdomen are segmented. So
D.
D, of course. It's only the abdomen, طبعا, not the cephalothorax خالص. Number B, no. Go to 5. Cephalothorax and abdomen not segmented, means all the rest. Abdomen and cephalothorax about the same size. So let's go here.
Abdomen, cephalothorax about the same size. F.
F, of course. Um, Abdomen larger than the cephalothorax, body covered with long hair, and abdomen is larger than the cephalothorax. Well, body is covered with long
C.
hair, of course, it's the only one. And the last one, body not covered with hair, but the abdomen is larger than the cephalothorax. So it's going to be
E. I mean B. I mean B.
B, B, yes.
Yeah, I did not see that. This
Cephalothorax means like,
question deserves 10 marks.
it's only 4. لا
Yeah,
حرام.
it's too much.
No, not this one. There's no. 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 I think that's all for this part, because they are not putting them in order in the old tests.
So you have to search
I
for
have
them.
to search. But let me go for, but I found that they repeat the questions, copy and paste.
I realized that from the multiple choices we solved a lot about
Not
the
only the
sensitivity,
multiple, yes,
sensitivity.
not only the multiple choice.
Yeah, also the paper for.
Yes. So not this one. Yes, this one is good. So reptiles and mammals are both vertebrate state. 3 features for mammals that are not found in reptiles.
From the picture, or can we just say?
No, 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 no. They didn't say from the picture.
Okay, so hair or fur, we
Mhm.
can mammary glands also,
Yes, very good.
and the external ear.
Very good. Very good. Okay. How some mammals are able to stay now. I think that's it. That's it. Okay. Good. Yani, you are doing good. Let's return back to our revision. Now we are going to begin chapter 3, which is about movement in and out of the cell, of course. Now we have uh, two ways by which the molecules or the particles or materials, they move in and out. Two ways. One of them is a passive way or a passive method. It means it does not need energy. And the other method is an active method that will need energy. Now let's begin by this passive method. We have two of them. One of them for particles, and this is called diffusion, while the other one is for water, which is called osmosis. The active method, of course, is called the active transport. So the passive, without the need of energy, for particles, it is diffusion, and for water, it is osmosis. Now let's begin by diffusion. Diffusion means that you have to mention three things. If you are asked about the definition of diffusion, you have to say that particles move from high concentration to low concentration.
Against concentration gradient.
Not against, along,
Down, the
or
concentration gradient.
down, yes, the concentration gradient. So if you are asked about the definition of diffusion having three marks, you have to put this is one mark, this is another, and this is the third. Sometimes the students, they just mention these and they say, I got the full mark. No, you
You
didn't.
lost the mark.
You lost the mark. You have to say along or down concentration gradient type. I just said that this is a passive mechanism. It means that it does not need energy, but there is a certain energy that allows the particle to move from high to low concentration. Passive movement means that it does not need energy from the cell or from respiration. As I told you now a few minutes ago, that mitochondria, they are responsible for respiration to produce energy. In passive mechanism, no energy from respiration is taken, but the energy here, it comes from the particles themselves. So what type of energy? It is kinetic energy. And of course, we know from physics that kinetic energy is the energy resulting from movement. What is moving here? It is the molecules, because we know that the molecules, they are in continuous movement. And when they move, they bump together or they collide together. And this collision will let the molecules to return back with higher speed. So whenever the molecules are found together in a certain medium and they are colliding together or hitting each other. When they hit, if this molecule, for example, collided with this one, it's going to return back with a higher speed than its original speed. That's why during collision of these molecules together, they will gain more and more kinetic energy. That's why they can pass from high concentration to a low concentration. But here, there is a very important thing that you have to know, that all the students, they used to say through a partially permeable membrane. This is not correct. Permeable membrane or through a cell membrane. This is not correct, because gases, they are particles, they are molecules, and they can diffuse without the presence of a membrane. Yani, most of the students, they say that if this is a membrane, so the particles can pass from high concentration to low concentration through or across this cell membrane, or the opposite. If this is high and this is low, they are going the opposite way across a cell membrane. But what about gases? Yani, if you are standing in a room, in a corner of a room, and you have perfume with you, or eau de toilette or whatever, and you spray it, after a minute, you are going to smell this perfume all over the room, because the gases, they diffused all over the room without the presence of a membrane. For gases, they can diffuse without the presence of a membrane. But for other particles like salts, for example, sodium ions, potassium ions, chloride, nitrates, whatever, they have to cross a cell membrane or a partially permeable membrane in order to diffuse. But for gases, it's not a must. And by the way, when the gases diffuse through a partially permeable membrane, they have to dissolve in water. But if there is no membrane, gases can diffuse, of course, from where you are standing and you sprayed the perfume where the concentration of the perfume was high, while in the rest of the room there was no perfume at all. After a few minutes, the gases will diffuse throughout the whole room to fill, and you can smell the perfume anywhere. This is an important exam hint, that if you have a multiple choice question and they said that all particles need a membrane or a partially permeable membrane to diffuse, this is a wrong answer, because they say all. So never choose this. You have to choose that the gases, they do not need a partially permeable membrane in order to diffuse. طيب, فالدiffusion of gases and any solute is very important for the life of the cell, and of course for our lives. يعني there are materials that have to enter the cell or to be formed or to diffuse inside, for example, the mitochondria, in order to perform photosynthesis, like glucose, like oxygen. They have to diffuse inside the cell in order to give us energy by respiration. That's why I just said that diffusion of particles is very important to our life. And this is an example, diffusion of glucose from, of course, the blood to the cells, because if this is a cell and blood capillary is carrying blood, and of course we know that inside the blood we have a red blood cell that is carrying oxygen, and of course plasma is carrying glucose. So both glucose and oxygen, they are going to diffuse inside the cell in order to allow the cell to respire, to perform respiration. because respiration is glucose plus oxygen. This will give me energy together with, of course, carbon dioxide and water. So without this diffusion, we cannot stay alive. And this is one example. Like, are there factors that enhance or that makes diffusion better or faster or facilitates diffusion? Yes, there are. So factors that affect diffusion or allow diffusion to be better. Now, if you have, for example, a cell, and this cell, it has a cell membrane. This is very
Thickness of the
thin.
cell membrane or something.
Yes, of course. And I have here the capillary. The capillary wall is formed of only one cell. It means that this is one cell, and this is only a cell membrane. So the distance traveled by the particles is very thin. So particles can move to the cell or out of the cell to the blood very easily because the short distance that is traveled by the particles. The second thing is there must be a concentration gradient. And whenever this concentration gradient is more, so the easier is the diffusion. Let me give you an example. Yeah, of course, it's not a true example. Suppose this is a cell membrane, and I have here, for example, 100 sodium ions. On the other side of the membrane, I have 50 sodium ions. Of course,
It will pass in a minimum rate.
it will pass. Yes, this is diffusion. Suppose I have here 100 sodium ions.
The least it is, the more faster it will go.
Yes, and here I have only 2. So here it's going to be very fast, because here the concentration gradient is lower than the concentration gradient here. So whenever there is more concentration gradient, or we call it a steep concentration gradient, so diffusion will be better. The third factor is temperature. Whenever there is more temperature, of course, not to the extent that the cell becomes burned, becomes damaged. No, when there is more temperature, so this will lead to better diffusion. Why?
Molecules collide more often.
Very good, very good, excellent. These are 3 factors, and there is another one, which is
The size.
yes, but we say the surface area.
Area.
The
The greater the surface area, the faster the total diffusion.
Yes, of course. عشان إن شاء الله next year أو in the AS مش next year but we are going to begin next month إن شاء الله. You are going to see that there is something called surface area to volume ratio that is going to work as well with the diffusion, or to be added as a factor for diffusion. Now, this is for the particles. What about for water? And I just called it osmosis. Now, again, osmosis is a passive process that does not need energy, and it is specific for water. But here we say 3 components. High water what? From high water, excellent. Never say concentration. In OL we do not count it in the correction. لكن طبعا in higher grades you are going to be accused for this if you wrote concentration, not potential. To lower water potential down a water potential gradient. And again, this is not an active process, and it depends as well on the kinetic energy between the water molecules. فالosmosis it allows the water molecules to move across the cell membrane. Suppose I have a cell here, and inside, of course, there is a cytoplasm that has a certain concentration. Because I just told you that cytoplasm is a solution that contains water and the solutes are dissolved in it. If the amount of solute is high, so the concentration of the cytoplasm will be high. If the amount of water is more, so the concentration will be less. So let us here agree for something. If we have high solute and low water, so this is called a concentrated solution. Now, if I have the opposite, I have low solute and or high water, we say that this is a dilute, yes. But suppose I have the same concentration, so this concentration is the same. But if I have, for example, a certain concentration inside the cell, and in the medium outside, I have the same concentration. It means that the amount of water here will be equal to the amount of water here. Will water pass from either sides or no passage?
No passage.
No passage. It means that if I have a cell and water is not passing, so no change in size of the cell. And we say that because the concentrations are equal, we call this the isotonic concentration, or the concentration is isotonic. Suppose I have a cell here, there is a certain concentration inside and a higher concentration outside, means the amount of water outside is less than the amount of water inside. What's going to happen to this cell or to water?
Can you repeat? I was writing isotonic.
Oh, okay. If I have a cell and it has a higher amount of water than outside, what's going to happen?
Can pass through.
It can pass from inside out or from outside in?
Inside out.
Inside out,
Like
yes.
the water, water outside will pass in and the water inside will pass out.
Will pass out. Very good. What will happen to the cell? If this is an animal cell?
It will burst.
When the water gets out, it's not going
No,
to
it will,
burst.
what do you call it? It will just die.
What's the shape of the cell? It will
I have no idea.
shrink. We always use this word.
Right.
Where, by the way, if it is a blood cell, we call it crenation. A red blood cell, we call it crenation. We used to use this word in the old syllabus, but the new syllabus, they just write it shrinks. But what if I have a plant cell? If I have a plant cell like this, this is the cell wall, and this is the cell membrane, which is very near to the cell wall. And here I have the cytoplasm with high water content, and here outside there is low water. What happens to the plant cell? What happens to water at the beginning?
When the water is out, inside is more than outside?
Yes.
Nothing happens. The cell is firm.
No. Water inside is more than outside. What about osmosis?
The water won't pass through.
No, it will.
The water inside will go outside.
Will go outside. So what will happen to the cell?
What is the water switching or what?
No.
Then why does the water pass out?
The water will pass out. What will happen to the Cell membrane
It
and the
will.
cytoplasm. It
Ah,
has a name.
cytoplasm will go because the water is going out.
Yes, يعني you are going to see something like this. This is the cell wall. Cell wall is not affected, but the cell membrane
Oh, I
or
understood. I understood. I understood. I understood.
will
I
be
understood
like this.
now. Yeah,
Okay.
yeah.
What is this called? This shape. It is called plasmolysis.
Plasmolysis.
Yes, and we say that the cell is plasmolysed.
Realized.
Okay, this happens in the plant cell, but in the animal cell, we just say it shrinks because there is no separation here, the plasmolysis, because the cytoplasm lost its water, so the cell membrane separated from the cell wall. Remember when I told you that the cell
You
membrane.
cannot distinguish between the cell
Yes,
wall and cell
but
membrane
but why? Why we cannot distinguish? Because
because
the
they are very near
cytoplasm.
to each other.
Yes, but why again? Because the cytoplasm is pressing on the
Against.
cell membrane, keeping it
In
very near.
close contact with the cell wall.
Yes, but here the cytoplasm lost its water, so there is nothing to keep the cell membrane stuck to the cell wall, so it's going to be plasmolysed like this. Now, here we say that the water outside is hypertonic to the cell. Hypertonic means highly concentrated. طب why the water, the solution outside is highly concentrated? Because water is low. And I just mentioned this here. When there is high concentration, if the solute is more or if the water is low. Here I have low water. It means that this is a highly concentrated solution outside the cell, and we call it hypertonic to the cell. Here it was isotonic to the cell. The word iso means the same tone or the same concentration. Here it is hypertonic. The water outside is hypertonic to the cell. You got that? You got it?
Yes.
Okay, so
Yes.
let's go back to the opposite. Now here I have a cell, animal cell, and I have here high concentration, or in other words, low water, low water than outside. What's going to happen to water?
The water will go out of the cell outside.
Look carefully at the picture. Which part is heavy?
Okay. The water potential inside the cell is low, right?
Inside the cell is low, yes.
Okay, so the water will go inside the cell.
Go inside. But this is an animal cell. It
Yes,
does
it
not
has no
have,
cell wall, so it will burst.
it's going to burst. And we say that the liquid or the medium outside the cell is hypotonic. Hypotonic means of lower concentration, because of course the high water means diluted. So it is hypotonic. But what about the plant cell? If here we have low water and outside we have high water, what will happen now?
The water will go into the cell
Yes,
and will add pressure, but due to the cell wall, the cell will become turgid.
excellent. Now I just told you turgidity. I just told you that two things, or two structures in the plant cell, they share together in this turgidity, which are the cell wall and the vacuole. The vacuole, because it becomes full of water, because water, of course, is going to enter to the cytoplasm, and the vacuole will be filled as well with water. So this vacuole is going to press on the cell wall, helping the turgidity of the plant cell. طبعا this is going to be to a certain extent. يعني suppose there is more and more water imbibed or passing to the plant cell, so at a certain time the cell wall will not be able to withstand even because so it's going to rupture, of course, or to burst. This is the passive kind of transport across a cell membrane. Let's go now to the active transport. Active transport, from the name that this is an active mechanism, it means it needs energy. But here the energy does not come from the particles, does not come from the collision of particles. It comes from the energy of respiration. Yani when we respire, part of the energy is responsible for this active transport. Right. If I want to describe active transport, I have to mention three components. The particles will move from low concentration to high concentration
against a concentration gradient.
Very good. Concentration gradient. And I will always explain this to my students. If I have here a high road, for example, or a surface or whatever, or a mountain, and somebody is standing here. And this is the mountain. Just a second to get down. Somebody is standing up the mountain, or the hill or whatever, and they want to go down. This is high concentration, and here is the lower. So they are going to move very fast without any energy. Just their legs are going to move. But suppose the opposite. This person is standing at the low level, and they want to climb the mountain. They are going to make an effort, energy in order to rise or to climb against the gradient, because of course this is a gradient difference between two heights. This is the active transport, and we always use this active transport, for example, the root hair cells. And in our villi of the small intestine. Yani for root hair cells, suppose this is the soil, and here are the soil particles, and here are, for example, the ions dissolved in water. Now this is the root, and this is the root hair cell. I'm going to draw only one. So inside the root hair cell, there is a high concentration of particles, higher than outside. So the particles will move against the concentration gradient in order to be absorbed to the inside of the root hair cell. What about the villi of our intestine? Suppose I have here the small intestinal wall, and this is the villus, and inside, of course, there are blood vessels, blood capillaries. In the blood, for example, and this is the food, digested food. Suppose I have here glucose. After digestion of starch, طبعا, it's going to produce glucose inside the small intestine. In the blood, glucose is higher than the lumen of the intestine. So because glucose is very much needed, it's going to pass by active transport from the lumen of the small intestine to the blood. These are two examples that show you how the active transport is needed for both animals and plants.